History of vaccination. The story of vaccines did not begin with the first vaccine used by Edward Jenner. Jenner used a material from cowpox pustules to provide protection against smallpox. It all begins with the long history of infectious disease in humans. In particular, the early uses of smallpox material to provide immunity to that disease. The practice of immunization dates back hundreds of years. Buddhist monks drank snake venom to confer immunity to snake bite. Variolation is smearing of a skin tear with cowpox to confer immunity to smallpox. It was practiced in 17th century China. Evidence exists that the Chinese employed smallpox inoculation or variolation as early as 1000 CE. It was practiced in Africa and Turkey as well, before it spread to Europe and the Americas. Abu Bakr Muhammad Zakaria Razi, known as Razis in the Western world, was a Persian physician. He was the first to publish a written account titled On Smallpox and Measles. In that he describes the two diseases as being distinct and unique from each other. Several accounts from the 1500s describe smallpox inoculation as practiced in China and India. Glynn and Glynn, in The Life and Death of Smallpox, note about Emperor Kang as of late 1600s. He had survived smallpox as a child and had his children inoculated. This method involved grinding up smallpox scabs and blowing the matter into nostril. Inoculation may also have been practiced by scratching matter from a smallpox sore into the skin. It is difficult to pinpoint when the practice began, as some sources claim dates as early as 200 BCE. During 1545, around 8,000 children died in Goa, India, from a smallpox epidemic most likely introduced by the Portuguese. During 1578, Guillaume de Bellu described an epidemic of pertussis, whooping cough, in Paris. He referred to it as quint, which he said was a common name for the disease that was circulating. De Bellu suggested the name might have to do with the sound of the characteristic whoop cough. 1612, Henry Prince of Wales, the oldest son of King James I, died at age 18 after a short illness. It was not identified or described other than as a fever. In 1882, Norman Moore, M.D., published The Illness and Death of Henry Prince of Wales in 1612. He studied of the autopsy on the prince as well as detailed descriptions of the illness. Moore alleged that the prince had died of typhoid fever. If it was correct, this would have been the earliest English case of typhoid fever on record. Strangulations. 1613 was the year known in Spain as El Año de los Garrotillos, strangulations, for its epidemic of diphtheria. During 1625, early smallpox in North America. French Jesuits who encountered Native Americans in Canada were confused by the hostility showed by the Indians. Indians, they said, observed with some sort of reason that since our arrival in these lands, those who had been the nearest to us had happened to be the most ruined by smallpox, and that whole villages of those who had received us now appeared utterly exterminated. Year 1633 was that of colonial epidemic. A smallpox epidemic hit Massachusetts, affected settlers and Native Americans. Among the casualties were 20 settlers from the Mayflower, including their only physician. Quarantine in Boston declared during 1648. There was an epidemic of yellow fever in Barbados, Cuba, and the Yucatan. A strict order for quarantine was established in Boston, Massachusetts. It quarantined all ships arriving from the West Indies because of U plague or like infectious disease. Measles appears in Boston in the year 1657. In Boston, John Hull wrote in his diary that the disease of measles went through the town. But fortunately there were only very few deaths. Bladders in the windpipe, 1659. Cotton Mather, 1663–1728, a Boston minister, wrote, in December 1659 the, until then unknown, malady of bladders in the windpipe, invaded and removed many children. By opening of one of them the malady and remedy, too late for very many, were discovered. He was most likely describing diphtheria. One family afflicted was that of the Reverend Samuel Danforth. Danforth wrote, "The Lord sent a general visitation of children by coughs and colds. of which my 3 children Sarah, Mary and Elizabeth Danforth died, all of them within the space of a fortnight. Royal support of inoculation announced in 1661. When Chinese Emperor Fu Lin died of smallpox, his 3rd son became Emperor Kang. He had survived a case of smallpox before he became emperor. He eventually supported inoculation and wrote about it in a letter to his descendants. Measles. Sydenham documents measles infection. The Persian physician Rhazes was the first to attempt distinguishing smallpox from the measles. But Sydenham was the first to do so successfully and in detail. He had also recorded the details and distinguished the disease from scarlet fever. The Indian plague breakout during 1679. French courtier Count de Frontenac Louis de Bois described the effects of smallpox on the Iroquois. He termed it the Indian plague and continued, "The smallpox desolates them to such a degree that they think no longer of meeting nor of wars, but only of bewailing the dead, of whom there is already an immense number." A royal death, 28th of December 1694. Queen Mary II of England, aged 32, died of variola hemorrhagica, a lethal form of smallpox, during which bleeding occurs into the pustules, from other body surfaces, and internally. The havoc of the plague had been far more rapid. But the plague had visited our shores only once or twice within living memory. And the smallpox was always present, filling the churchyards with corpses, tormenting with constant fears all whom it had not yet stricken, leaving on those whose lives it spared the hideous traces of its power, turning the babe into a changeling at which the mother shuddered, and making the eyes and cheeks of the betrothed maiden objects of horror to the lover. Toward the end of the year 1694, this pestilence was more than usually severe. At length the infection spread to the palace, and reached the young and blooming queen. The African use of variolation was noticed during 1706. Cotton Mather, a Boston minister, 1663 to 1728, received a gift of a Libyan-born enslaved person named Onesimus. He bore a scar from smallpox variolation in Africa. Mather inquired among other enslaved people and found that many had been variolated. They thought themselves as immune to the disease. Later, Mather would read of variolation in English medical journals. He started to promote the practice in Massachusetts. Deadly toll. Deadly toll for prominent colonists took place in 1713. Measles killed 3 of Cotton Mather's children and his second wife. Mather, a prominent Boston clergyman, would later bring smallpox variolation to the colonies. But in 1713 he wrote in his diary of an approaching measles epidemic. "The measles coming into the town, it is likely to be a time of sickness and much trouble in the families of the neighborhood." In short order, as the rest of the town suffered, measles infected most of his family members. It killed his wife, his newborn twins, another daughter, and the family's maid within a few weeks. Variolation in Turkey was done in 1718. In 1718, Lady Mary Wortley Montagu, 1689 to 1762, had her son variolated in Constantinople by Dr. Charles Maitland. Lady Montagu, whose husband was ambassador to Turkey, had been disfigured by smallpox around 1715. She had heard about variolation upon her arrival in Turkey, and was anxious that her 6-year-old son, Edward, have the procedure. Boston smallpox epidemic. Smallpox raged through Boston in 1721, ending in 844 deaths. During this epidemic, Cotton Mather urged the physician Zabiel Boylston to variolate. He variolated 248 people, thereby introducing variolation to the Americas. Of those variolated, 6 died. The case fatality for variolation was about 3%, and the disease case fatality was 14%. About 900 people left town for fear of catching the disease. At Harvard, the chambermaid of Cotton Mather's son Samuel contracted smallpox. Samuel's brother Increase encouraged his father to have Samuel variolated by Boylston. Samuel too had survived the procedure. Mather was widely criticized for his role in promoting variolation. A primitive grenade was thrown through a window of his house. The attached note threatened Cotton Mather, "You dog, damn you. I'll inoculate you with this, with a pox to you." First English variolation. The first English variolation was done on 21st of April, 1721. Lady Mary Montagu brought the practice of variolation to England. She made Dr. Charles Maitland to variolate her 2-year-old daughter. Lady Montagu would come under considerable criticism for advocating variolation. But this practice slowly began to spread as its ability to protect against smallpox became apparent. The results, however, were sometimes fatal. 2 to 3% of those who died of smallpox, in contrast to 20 to 30% who died after contracting smallpox naturally. What's more, variolated individuals could pass the disease on to others. The plague among children spread during 1735. A terrifying diphtheria epidemic swept through New England. In some cases, entire families died of the disease. In one New Hampshire town, 32% of children under 10 died of diphtheria. The case fatality ratio was almost 40%. Noah Webster, 1758 to 1843, later wrote, "It was literally the plague among children. Many families lost 3 of 4 children, many lost all." Treatments were largely ineffective. Franklin loses his son on 21st of November 1736. Benjamin Franklin's 4-year-old son, Francis Folger Franklin, died of smallpox. Rumors began to circulate claiming that the boy had been inoculated. Franklin published a denial and advocated inoculation. The German measles, 1740, rubella. German physician Friedrich Hoffmann was the first to give a clinical description. It was about a disease that would later come to be known as rubella. A new name for a disease, 1741. The term yellow fever began to be used in the 1740s for the disease we now know by that name. Doctors had referred to the disease, and others that were similar to it, by a variety of names. They include pestilential fever, malignant fever, and putrid bilious fever. Yellow fever. The name comes from the yellowed appearance of the skin and eyes. Those results from damage to the liver. Toxic materials build up in the blood and cause the telltale color to appear. Philadelphia avoids an outbreak in the year 1747. Philadelphians were aware that yellow fever had been traced to the West Indies. A vessel arriving in the city from Barbados was examined carefully. The captain reported the death of a sailor as well as a second possible case of yellow fever. The ship was held in anchor more than a mile from the city and prevented from unloading cargo or passengers. Infectious nature of measles shown, 1757. Scottish physician Francis Home, MD, transmitted measles from infected patients to healthy individuals via blood, demonstrating that the disease was caused by an infectious agent. Harvard, Adams, and smallpox, 1764. A smallpox outbreak in Boston led the General Court to flee across the river to Cambridge. Unfortunately, they required such roaring fires in the fireplaces of Harvard Hall that the whole building burned down, destroying the best collection of scientific apparatus and the finest library in the American colonies. The fire took nearly the entire college library with it. One book, which had been taken from the library by a student earlier in the evening, survived. John Adams, 1735 to 1826, underwent variolation during a Boston epidemic. By this time, many doctors performed variolation along with an arduous course of purging, vomiting, and sweats brought on by treatments with mercury and antimony. Chickenpox disease distinguished from smallpox, 1767. English physician William Heberden was the first to give a detailed description. In that, he distinguished chickenpox from smallpox. He wrote, "These pox break out on many without any illness or previous sign. In others they are preceded by a little degree of chillness, lassitude, cough, broken sleep, wandering pains, loss of appetite and feverishness for 3 days. Most of them are of the common size of the smallpox, but some are less. I never saw them confluent nor very numerous. The greatest number which I ever observed was about 12 in the face. and 200 over the rest of the body. Chickenpox. Heberden noted that those who had previously chickenpox were not capable of having it again. Smallpox as war breaks out, 1775. George Washington's siege of Boston was complicated by smallpox infection inside the city. British troops occupying Boston had been variolated or exposed to smallpox in the past. But Washington's Continental Army troops were more vulnerable. Most had never been exposed to smallpox and were not previously variolated. Washington himself had survived a case of smallpox while on the island of Barbados in 1751. During 1776, smallpox becomes a weapon of war. Of a force of 10,000 Continental Army soldiers in Quebec, about 5,000 fell ill with smallpox. A British commander may have deliberately intended to spread the disease. He had sent recently variolated civilians into the Continental Army encampments. The Continental Task Force commander, Major General John Thomas, died of smallpox. The unit retreated southward in May 1776. Arguably, this defeat preserved the status of the northern British colonies. It allowed Canada to become the separate country as it is today. John Adams wrote, "Our misfortunes in Canada are enough to melt the heart of stone. The smallpox is 10 times more terrible than the British, Canadians and Indians together. This was the cause of our precipitate retreat from Quebec." 1777 was the year of mandatory inoculation for Continental Army. George Washington was the commander-in-chief of the Continental Army. Based at his headquarters in Morristown, New Jersey, he had ordered mandatory inoculation for troops. It applied to those who had not survived a smallpox infection earlier in life. This was possibly in reaction to the inability of Benedict Arnold's troops to capture Quebec from Britain the year before. During that war, more than half of the colonial troops had smallpox. Recruits passing through Virginia were inoculated at Alexandria. Edward Jenner's innovations, begun with his successful 1796 use of cowpox material to create immunity to smallpox. It quickly made the practice widespread. Edward Jenner. Jenner's breakthrough, 14th of May, 1796. Edward Jenner tested the hypothesis that infection with cowpox could protect a person from smallpox infection. Cowpox is an uncommon illness in cattle, usually mild, that can be spread from a cow to humans via sores on the cow. During an infection, dairy workers may have pustules on their hands. Sufferers can spread the infection to other parts of the body. We know now that the cowpox virus belongs to the orthopox family of viruses. Orthopox viruses also include horsepox virus, monkeypox virus, and variola virus, which causes smallpox. On May 14, 1796, Jenner inoculated 8-year-old James Phipps with matter from a cowpox sore on the hand of milkmaid Sarah Gnomes. Phipps suffered a local reaction and felt poorly for several days but made a full recovery. In July 1796, Jenner inoculated Phipps with matter taken from a fresh human smallpox sore, as if he were variolating the boy, in an attempt to challenge the protection from cowpox. Phipps remained healthy. Jenner next demonstrated that cowpox matter transferred in a human chain, from one person to the next, provided protection from smallpox. Jenner was not precisely sure about the nature of the cowpox material he used. He suspected that cowpox actually came from horsepox, in other words, he speculated that cows became infected with the same agent that caused a similar disease in horses. Recent genetic analysis of old samples of smallpox vaccine have revealed that the samples were more closely related to horsepox virus than cowpox virus. His method underwent medical and technological changes over the next 200 years. It eventually resulted in the eradication of smallpox. More than the science behind vaccines, these timelines cover cultural aspects of vaccination as well. It starts from the early harassment of smallpox variolators to the establishment of vaccination mandates, to the effect of war and social unrest on vaccine-preventable diseases. Edward Jenner, Louis Pasteur, and Maurice Hilleman are pioneers in vaccine development. Edward Jenner was the first to test a method to protect against smallpox in a scientific manner. He did his study in 1796, and although he did not invent this method, he is often considered the father of vaccines. It was his scientific approach that proved that the method worked. The method Jenner tested involved taking material from a blister of someone infected with cowpox. It was inoculated into another person's skin. This was called arm-to-arm inoculation. Edward Jenner is considered the founder of vaccinology in the West in 1796. He inoculated a 13-year-old boy with vaccinia virus, cowpox, and demonstrated immunity to smallpox. Smallpox. In 1798, the first smallpox vaccine was developed. 1800 Waterhouse brings vaccination to the States. Benjamin Waterhouse, 1754–1846, a Harvard professor of medicine, performed the first U.S. vaccinations on his children. He went on to put much effort into encouraging public vaccination. Russian Empress Dowager encourages vaccination, 1801. She named an orphan first given the smallpox vaccine Vaccinov. She provided the young girl with a pension for life. Vaccination versus variolation, 1802. Dr. Jean de Caro, physician, indirectly helped to introduce vaccination to India. He suggested that inoculation should be eliminated, saying, "It is inconsistent for a government to encourage vaccination and not forbid inoculation." This was among the first of many efforts to encourage vaccination over variolation. In 1802, Massachusetts became the first U.S. state to encourage the use of vaccination against smallpox. In 1805, Mary Annalisa of Lucca, Napoleon's sister, became the first ruler to try making vaccination compulsory. She was unable, however, to determine a practical method of enforcement. 1810, Italian physician Gennaro Galbiati began retro-vaccinating cows with human vaccine lymph. He then vaccinated people with the resulting animal lymph. Beginning in 1817, a series of deadly cholera pandemics swept over India, Asia, and the rest of the world. Scholars usually refer to a wave of 7 cholera pandemics, and generally describe them as occurring. 1817-23, 1826-37, 1846-63, 1865-75, 1881-96, and 1902-23, and 1961 present. 1826, diphtheria, which had previously been called by a variety of names, gained its official name. French physician Pierre Bretonneau, 1778–1862, called the disease diptheria. The origin was the Greek word for leather or hide. It describes the coating that appears in the throat, that is, the pseudomembrane. Bretonneau also distinguished diphtheria from scarlet fever. 1831, as a schoolboy in Arbois, France, Louis Pasteur observed, "At first hand a rabies outbreak spread by the bites of rabid animals." Edward Ballard. 1836, English physician Edward Ballard noted that cowpox transmitted from human to human seemed to decline in potency over time. He recommended choosing new strains of cowpox and reintroducing the pustule matter, lymph, back into cows to boost its potency. This method came to be used to supply sufficient material for vaccination. 1841, rubella, the naming of the Little Red. The name rubella, meaning Little Red, was first used to describe an outbreak at a boys' school in India. 1854, cholera, Pacini links bacterium to disease. Italian physician Filippo Pacini, 1812–1883, linked the cholera bacterium to the disease itself. Pacini microscopically observed samples from the intestines of cholera victims. He noted the presence of tiny, comma-shaped particles that he suggested were the cause of the disease. The bacterium is known today as Vibrio cholerae. The genus Vibrio includes several species of comma-shaped or rod-shaped bacteria. They are mobile by means of a whip-like flagellum, or tail-like structure. Pacini expanded on his ideas about cholera in several later publications. He suggested that the cholera vibrio acted on the lining of the intestine to cause massive fluid and electrolyte loss. He suggested that cholera patients be treated with intravenous injections of water with salt added. Though correct, the scientific world largely ignored his work during his lifetime. 31st August, 1854, cholera, the pump handle. John Snow once again made an important contribution to the understanding of cholera. Snow proposed that the water, drawn from a downstream Thames River, was contaminated with the causative agent of cholera. 1857, The Biology of Fermentation. Louis Pasteur was named Director of Scientific Studies at a prestigious education institution in Paris. At the École Normale, Pasteur continued his studies of fermentation. He established the concept that fermentation was a biological process, carried out by tiny organisms. Importantly, his experiments demonstrated that fermentation and rotting could not take place when the organisms were excluded from solutions by heating or by filtering. Pasteurization. His ideas would lead to the widespread adoption of what we call pasteurization for the preservation of food, and to increasing attention in surgery to the role of microorganisms in infection. This concept of the activity of microorganisms emerged further in the study of the causes of infectious disease later. 10th of September, 1859, Pasteur's daughter Jean, age 9, died of typhoid fever. Measles plays a role in the Civil War, 1861. Measles sickened troops on both sides of the American Civil War. 19th of November, 1863, Lincoln contracts smallpox. Hours after delivering the Gettysburg Address, President Abraham Lincoln suffered weakness, fever, and headache on the train back to Washington. Within a few days he developed widespread pustular lesions. Medical historians assert that Lincoln had smallpox, which sickened him for almost 4 weeks. Lincoln, however, recovered, though his valet caught the disease and died. 11th September, 1865, Louis Pasteur's 2-year-old daughter Camille died of a liver tumor. 23rd of May, 1866, Pasteur's daughter Cécile, age 12, contracted typhoid fever and died. 19th of October, 1868, Pasteur suffered a cerebral hemorrhage. He was treated at home, initially with the application of 16 leeches behind his ears. His family and colleagues feared that he would not survive, but he slowly recovered. His left arm and leg were permanently paralyzed. 1878, yellow fever killed more than 13,000 people in the lower Mississippi Valley. At the time, it was one of the worst medical disasters in U.S. history. 1879, Louis Pasteur produced the first laboratory-developed vaccine. The vaccine for chicken cholera, Pasteurella multicida. 14th August, 1881, Carlos Finlay, 1833–1915, presented a paper. The mosquito hypothetically considered as the transmitting agent of yellow fever. It was the first to correctly identify mosquitoes as the ultimate source of the disease. 24th of March, 1882, Robert Koch, 1843–1910, announced his discovery of the agent that causes tuberculosis. For a time, it was called Koch's bacillus. Today it is called Mycobacterium tuberculosis. Diphtheria. 1883, Edwin Klebs, 1834–1913, a Swiss-German pathologist, identified and described the bacterium that causes diphtheria. It was known at first as the Klebs-Leffler bacterium. The bacterium's club-shaped appearance helped Klebs differentiate it from other microbes. Later, the bacterium became known as Microsporon diphtheriticum, Bacillus diphtheriae, Mycobacterium diphtheriae, and, as it is called now, Corynebacterium diphtheriae. 1884, Leffler's contributions. Friedrich Loeffler, 1852–1915, a German bacteriologist, was the first to cultivate Corynebacterium diphtheriae. Loeffler used a set of rules we now know as Koch's postulates to confirm that Corynebacterium diphtheriae was the agent that caused diphtheria. Later in 1884, Loeffler showed that C. diphtheriae produces a toxin. This was the first description of a bacterial exotoxin. An exotoxin is a toxin excreted into the surrounding environment by a microbe. When C. diphtheriae colonizes the upper respiratory tract, the bacteria secrete a toxin that injures and then destroys cells. Waste products and proteins form a thick gray substance called a pseudomembrane over the pharynx. The pseudomembrane sticks to tissues and may obstruct breathing. Meanwhile, the toxin may travel to the heart, muscle, kidneys, liver, and other areas. Koch's postulates. Robert Koch and Friedrich Loeffler's steps formed the basis for identifying a disease-causing agent. 1. The microbe is present in every case of the disease. 2. The microbe can be taken from the host and grown independently. 3. The disease can be produced by introducing a pure culture of the microbe into a healthy host. The microbe can be isolated and identified from the host infected in step 3. One exception to step 3 is that some individuals may be infected with a disease-causing microbe and not show signs of the disease. These are known as asymptomatic carriers. 2nd February, 1884, cholera, Koch isolates bacillus. Robert Koch, 1843–1910, in a letter from India describing his research there, announced his discovery of the agent that causes cholera. Tuberculosis. For a time, it was called Koch's bacillus, as was tuberculosis, and Koch's bacillus. Filippo Pacini had achieved this feat in 1854, but his work was not widely known. 10th of October, 1885, another success with rabies vaccine. Pasteur repeated his rabies vaccinations on a shepherd who had been severely bitten by a rabid dog. This episode, too, was successful. 1885, O'Dwyer uses intubation. American physician Joseph P. O'Dwyer, 1841–1898, introduced intubation for treatment of obstructed larynx in patients with diphtheria. Not only did O'Dwyer describe the method for the procedure, but he developed instruments to perform it. Within a short time, intubation largely replaced tracheotomy in treatment of diphtheria with airway blockage. O'Dwyer died a few years later from a heart ailment brought on by diphtheria infection, most likely acquired from a young patient whom he intubated. 1885 cholera, Ferran's vaccine. Spanish physician Jaime Ferran, 1852–1929, developed a live, attenuated cholera vaccine. His vaccine was the first to immunize humans against a bacterial disease. Louis Pasteur's 1885 rabies vaccine was the next to make an impact on human disease. 1890, antitoxin and serum therapy. Shibasaburo Kitasato, 1852–1931, and Emil von Behring, 1854–1917, immunized guinea pigs with heat-treated diphtheria toxin. Kitasato and von Behring showed that the blood products, sera, or singular, serum, of the guinea pigs contained a substance that prevented the harmful effects of C. diphtheria and its toxin. When the guinea pigs were re-exposed to lethal doses of the bacteria and toxin. Next, they showed that they could cure diphtheria in an animal by injecting it with the serum of an immunized animal. They called the substance antitoxin and their treatment serum therapy. They realized that they needed to immunize large animals, such as horses and sheep, to produce enough antitoxin to protect humans. Von Behring would win the first Nobel Prize in Medicine in 1901 for his work on diphtheria. 1892, Pfeiffer discovers bacterium. German physician Richard Pfeiffer, 1858–1945, once a student of Robert Koch, isolated a bacterium from the lungs and sputum of influenza patients during a pandemic. Influenza. Pfeiffer believed that he had found the cause of influenza, and the bacterium came to be known as Pfeiffer influenza bacillus. In the 1930s it was definitively established that influenza was in fact caused by a virus, not a bacterium. Pfeiffer's influenza bacillus, however, would eventually be named Haemophilus influenza, because of its long-standing, though incorrect, association with influenza. 1905, serum sickness described. Clemens Bierke, 1874–1929, and Bela Schick, 1877–1967, described a clinical illness they called serum sickness. Understanding this new form of disease paved the way for defining allergy and immunologic diseases. 1906, whooping cough, bacteria isolated. Belgian scientists Jules Bordet, 1870–1961, and Octave Gengu, 1875–1957, isolated the bacterium Bordetella pertussis. the causative agent of pertussis, whooping cough, which they had first observed in 1900. It would later also come to be known as the Bordeaux dengue bacillus. 1908 poliovirus identified. In Vienna, Karl Landsteiner, M.D., 1868–1943, and Erwin Popper, M.D., 1879–1955, announced that the infectious agent in polio was a virus. Over the 18th and 19th centuries, systematic implementation of mass smallpox immunization was done. It culminated in its global eradication in 1979. And then, at the dawn of bacteriology, developments rapidly followed. Antitoxins and vaccines against diphtheria, tetanus, anthrax, cholera, plague, typhoid, tuberculosis, and more were developed through the 1930s. Louis Pasteur's experiments spearheaded the development of live attenuated cholera vaccine in 1897. An activated anthrax vaccine in humans was developed in 1904. Plague vaccine was also invented in the late 19th century. Between 1890 and 1950, bacterial vaccine development proliferated. That included the Bacillus calmette-guerin, BCG, vaccination, which is still in use today. In 1923, Alexander Glennie perfected a method to inactivate tetanus toxin with formaldehyde. The same method was used to develop a vaccine against diphtheria in 1926. The middle of the 20th century was an active time for vaccine research and development. Methods for growing viruses in the laboratory led to rapid discoveries and innovations. That included the creation of vaccines for polio,